Hello, I'm Harry Glorikian, and this is the Harry Glorikian Show, where we explore how technology is changing everything we know about healthcare. If you're a parent, you've probably had this experience many times. Your young child has a fever and maybe a sore throat, but you don't know exactly what's wrong. Is it a bacterial infection? In which case an antibiotic might help. Or is it a viral infection? In which case you just have to wait it out. The symptoms of bacterial and viral infections are often the same, and most of the time, even a doctor can't tell the difference. Viral infections are more common, but sometimes the doctor will prescribe an antibiotic anyway, if only to help the parents feel like they're doing something to help. But what if doctors didn't have to guess anymore? What if there was a fast, easy blood test that a doctor could run in their own office to look for biomarkers that discriminate between bacterial and viral infections? Well, that's the seemingly simple problem that a company called MeMed has been working on for the past 13 years. Recently, MeMed's first testing product got approval from the FDA, and now the company is finally beginning to roll out commercially in the US. And here to tell us more about how it got built, how artificial intelligence fits into this picture, and how rapid diagnosis could change the practice of medicine is MeMed's co-founder and CEO, Eran Eden. MeMed has a growing office in Boston, but I reached him at his office in Haifa, Israel. Eran, welcome to the show.
Thank you very much for having me.
It's great to have you here. I know that there's a significant time difference, so I appreciate but it still looks like it's really bright and shiny out there right now. So what time is it in Israel right now?
5 o'clock in the evening.
It's 5 o'clock. All right. Yeah. Well, you guys have a lot more sun than we do anyway, because we're in the middle of winter. But
Absolutely.
So,
and the office here is actually full of people as well. So yeah, you don't stop innovation at 5 o'clock in the evening.
So, I was looking at your background, and I mean, it's interesting. It's diverse. You have a degree in biology, computer science, systems biology. Your first job was in computer vision, data analysis, but then all of a sudden you wound up starting a company that builds sensors and software for infectious disease. How how did you end up down this path? And do you feel like everything that you were doing until you got here was preparing you for it?
Well, I think a great question. So I think on the face of it, obviously the background in data science, as in molecular biology, obviously all of that relates to what we're doing as part of our day-to-day. And it is a good starting point. But in reality, there's a very big gap between what I was trained to do and today my everyday, day-to-day activity. I would say that probably the most important training that I got during my PhD at the Weizmann Institute has got less to do with differential equations or molecular biology, and it was more about a story that my mentor, Professor Uri Alon, told me when I was 3 years into the PhD. About 3 years into the PhD, he asked me, "Am I already in the cloud?" I said, "What?" And he said, "Are you in the cloud?" I said, "Well, what is the cloud?" He said, "Well, every PhD, every scientist, when you start your PhD, you go and read the latest papers in science and in Nature, and you see how somebody starts at point A, makes a hypothesis about point B, and then take the straight line from A to B. And then you say, okay, I'm going to do the same thing. And you start at point A, the known, you shoot for the unknown, and you start going, and suddenly you hit a roadblock. And then you hit another one, and another one. And at a certain point, you really lose direction, which he called the cloud. You're in the cloud. And then if you have enough perseverance and luck, you find a point C, which is not exactly where you thought you're going to end. You go there with, your last energy. And if you're lucky enough, then you publish another paper, how you started at point A, went from point C, and connected between the two dots with a straight line. And then you have another generation of PhDs that are asking themselves, well, why am I the only one that's struggling? And that lesson about how to be in the cloud, how to deal with uncertainty, to deal with failure, and still move on. That is probably more important in the training that I got to become an entrepreneur and CEO of a company than any specific scientific knowledge.
Yeah, no, I mean, trial and error, dusting yourself off, getting up and moving forward is, my wife calls me crazy when I keep doing it. But I think you have to be a little on the edge to constantly keep repeating and being willing to fail and then stand up and then move on. Maybe I think I was reading a paper recently that said forgetting quickly is a evolutionary, positive trait, so that you forget what happened that wasn't good and you keep moving forward. So, but let's talk about your company, MeMed. Like, you started that in, I believe it was 2009. And what was your founding vision? I mean, if you can talk about what you and your co-founder, did when you came up with this idea. I think you were both studying at the Technion at the time.
Yeah, so he was studying at the Technion, MD, PhD. I was studying at the Weizmann Institute in data science and biology. And frankly, I would love to tell you a story about a vision, but it started with a game. I don't think we had the presumptions to have really something that would grow to what MeMed actually became today. It was playing. We both had different reasons, first of all, for doing this. I can say that from my end, there was probably a pretty big gap between the places, the caliber of where we were able to publish high impact journals. And when I was looking at myself in the mirror and I was asking myself, is this actually going to have an impact on real patients? I couldn't really see the connection. There's another reason why I decided to found MeMed or co-found MeMed. That's probably off topic for today. We can take this on a beer sometime when we meet face to face. But so it's, first of all, it didn't start with a vision. It started with a scratch, wanting to apply some of the know-how that we had in converging between molecular immunology and data science, and to try to solve big, ugly problems that don't have a good solution in 21st century medicine, and trying to find something pragmatic. Now, rather than having it a eureka moment, I think, some entrepreneurs describe a eureka moment where suddenly you have the best and coolest idea and vision. For us, it was darkness for almost a year. Rather than a eureka moment, it was more like an evolutionary process, trial and error. We tried a bunch of solutions to problems that didn't really exist, until eventually we came up, with what we want to work with. But again, was no eureka. And the way that it actually started was, again, Kfir was coming from med school, talking about this problem of AMR, antimicrobial resistance, and the problem of distinguishing between bacterial and viral infections. And given our different background, we said, that's interesting. How can we apply immunology and data science to try to solve that? And at that point, we formulated what was to become MeMed's vision. And MeMed is based on a very simple premise, or a very simple idea. Our immune systems have evolved to tell us what's going on in our bodies. And all we do at MeMed is we listen to the immune response with biochemical sensors and machine learning and what have you, and we use that to translate or decode the immune system into insights that can potentially transform the way that we manage patients with acute infections and inflammatory disorders. The first problem we went after, because that's a very lofty goal, was potentially the most prevalent clinical indication on the face of this planet: a child with sniffles, or an elderly patient that coughs. Come to the doc, they have a fever. As a parent, you're many times hysterical. You're asking yourself, is it a bacterial infection or a viral infection? If it's a bacterial infection, antibiotics. If it's a viral infection, chicken soup. And I said, well, what if we can harness the immune system? What if we could measure or listen to the immune system in real time and use that to try to aid clinicians to tackle this seemingly simple problem? So the vision was listening to the immune response, and the first embodiment of the first problem we went after is this huge intractable problem: B versus V, bacteria versus viral infection, to treat or not to treat.
Yeah, I mean, it's funny you say simple, and I've worked in this area for a long time, and not not simple, not simple. But I've been watching dozens of companies over time try and tackle this problem, and everybody always comes at it and says, "Yep, we should be able to do it." And I'm like, "Okay, that's a big hill, to go and try and die on." So, but you got FDA approval for your device in the US, and I want to talk about that later. But it did take 13 years, which parts of the process turned out to be harder or slower than you thought it would be?
Before I answer that, I just want a minor correction. I didn't say it's a
Yes.
simple, I said it's a seemingly simple problem. In reality, it's an extremely difficult problem to go after. I think some of the biggest challenges that we have can be phrased in a very simple manner. But as you alluded to, yeah, it's an intractable problem. Bacterial and viral infections are often clinically indistinguishable. And it took us, over a decade to take this from an idea in a napkin in grandmother's kitchen. That's where we found, it was no garage, it was grandmother's kitchen, to what is considered a landmark FDA clearance that I think many folks did not believe we're going to be able to get this, because it required so many innovation, not only on the technological side, but also on the regulatory side. And when you ask, well, why only a decade? I think we're very lucky that it took us only a decade. And the challenge there, let's not call it a challenge. Let's call it problems. Challenges is something I always envy the people that have challenges. We have problems at MeMed, and we work every day to solve those problems, right? So there's many problems or hurdles you have to go through. So first of all, you have to overcome some pretty big research issues. Where do you find these hypothetical molecules of the immune response that go after bacteria and viruses? So research. Then you learn the hard way that research is very different from development. And development is very different than product. And product is very, very, very different than manufacturing. And manufacturing is very different than regulation. And regulation is very different than reimbursement and marketing, which is very different than commercial, et cetera, et cetera. So it's not good. It's not enough to excel in one thing. You have to really reinvent the wheel on several things. And as a company and as a team, reinvent yourself. And that's probably one of the biggest challenges. Your biggest impediment to progress is yourself and your team, because you might be an excellent data scientist, but you have to talk with a clinician. You might be an excellent clinician, but you have to talk the language of the molecular immunologist. You might be very versed in all of these three, but it's still not product, and it's still not the graphic user interface. And how is that connected to manufacturing? And really creating a culture or a team that can combine these seemingly very diverse elements within a small company, that is a very, very daunting and big task. And again, we frankly failed on multiple avenues there. We had to go back. We were in the cloud, and we had to reinvent point C again and again and again. So we were in a very far position than we are today, that we thought we were going to be at this stage.
So I'm going to, at some point, after this whole interview, I'm going to encourage you to write the next IBD book, because everything you said is absolutely the way that I've seen it over time. Is having to bring all these pieces together is not trivial in our work. But let's step back here for a second for everybody that's listening, right? Talk a little bit about basic immune system biology and the technology behind your diagnostic system. So if someone presents with an inflammatory response, why is it so hard for doctors to distinguish between the bacterial and viral infection?
Because bacterial and viral infections are clinically indistinguishable, and you don't have to be an MD to understand this intuitively. We know our kids so well, but still, when they have a fever or runny nose, we know that it's 80%, 85% a viral infection. But what if there's a lingering bacterial infection? And it turns out that because of the clinical manifestation is very similar, it's really hard to figure it out. not only children, also adults with say, a suspected LRTI or a fever without source. And even when we apply modern, I would say, diagnostics, there's still a big gap that remains. So, for example, when as a scientific community or a clinical community, when we approach this problem, we have tools at our disposal. Rapid strep tests, rapid influenza tests, multiplex PCR. In today's world, I think everybody, even my grandmother, is talking to me about the difference between rapid antigen tests. Suddenly becomes a really interesting topic over, over weekend dinners. Culture. So there's technologies out there, and going back to your question, why is there still a gap? And we've identified several things. The first one is probably most trivial, is time to results. Many of the clinical encounters, you want to have the solution here and now, whereas the technology that we have often provides solutions in hours and even days, and that's not always good. That's one hurdle, not the biggest one. The second one is that many times the infection site is inaccessible. Take, for example, otitis media, an ear infection, or sinusitis, or bronchitis, or pneumonia. It's really hard to reach the infection site and therefore identify the pathogen. It's 1 in 4 patients in the most prevalent disease on earth. That's really hard. Third, even if you use the best, most broad technology, diagnostics, to try to identify the bug, say a multiplex PCR, in more than 50%, 50% of the cases, you will not be able to put your hands on any microorganism. But you still, as a clinician, have to make a decision. And lastly, there's the issue of colonization. So even if you're lucky, the infection that is readily accessible, and you do get some sort of a virus, for example, that you detect, say, for example, an influenza, or let's take a rhinovirus. The rhinovirus is very prevalent in children. That's a problem. It's very prevalent in children. Even if you take seemingly healthy children, in a very high percentage of those children, they're going to have a rhinovirus. So mere detection does not imply causality. All this complexity is funneled into this few minutes that the clinician basically needs to make a decision, and it's a really hard dilemma because it's hard to know to distinguish between the two, and the ramifications could be quite significant.
So I know the answer to the question, but I'm going to ask it so you can explain it. So who cares? I mean, I know that it's ineffective to treat a viral infection with antibiotics, and that only work against the bacteria. But, we've been doing it trial and error. So what's the downside of doing that?
So it's actually it's a very deep question, because there's several layers. You're right. Sometimes people actually say there's several layers to answering this. The first one is, well, if you treat erroneously with antibiotics, because of this uncertainty, there's a lot of antibiotics overuse, that one of the consequences of this, it drives antimicrobial resistance, which basically means that the drugs don't work anymore. And if we continue on that path, we will potentially lose modern medicine, because if you lose the potency of antibiotics, you cannot treat infants or they have an infection, or a oncology patient that was succumb to a parasitic infection, or even have your wisdom tooth pulled out, because antibiotics won't be effective. And there's several quite influential studies that came out in the last few years. The last one actually in the Lancet came out 2 weeks ago, portraying a world without antibiotics, which is, we're seeing right now the consequences of COVID, of SARS-CoV-2. Some might argue it's not a smaller problem. So and it has both clinical and health economic consequences, according to Jim O'Neill, over $100 trillion by 2050 in lost GDP. That's not a trillion, $100 trillion. Yes, sorry, $100 billion. And it's a big number, right? It's a really, really big number. And maybe it's overly inflated, and maybe it's conservative, but it's a big problem. The issue is that nobody cares sometimes. The individual doesn't care, because the doctor right now, when he has a patient in front of him, he doesn't think about the masses. He thinks about the patient. So you might ask, well, why does the doctor care? Why does the patient care? And it turns out that there's an angle on the personal level as well, not only on the societal level. If you give erroneous antibiotics, you can drive anaphylactic responses. You can drive allergies, which have a toll. But then there's another element that people are less aware of. In addition to overuse, there's also simultaneously underuse. 1 in 5 patients that have a bacterial infection are not receiving antibiotics on time. There's much less publication on that, but it is a reality. And that also has consequences, including prolonged disease duration and sometimes even morbidity and mortality. So it's a really delicate equation, right? You don't want to overtreat, and you don't want to undertreat. Some countries overtreat, some undertreat. And again, at the end of the day, at the day-to-day, it does have ramifications both on the patient and on the doctor.
No, if we could accurately treat people, right, I think there would be a whole host of issues that could get solved and a whole host of issues that wouldn't emerge because of overtreatment or treating the wrong people. That, we could spend hours over a beer discussing microbiome and allergies and all sorts of other consequences of doing this. But so your system, which I love, is a basic blood test, right? So the MeMed BV looks at 3 immune system proteins in the blood: TRAIL, IP10, and CRP. So how are these proteins related to infection, and how can measuring their levels tell you about the nature of the infection?
Okay, so each one of those proteins that you just mentioned plays a critical role in the immune response to different invaders, bacteria, and viruses. What's special about this particular trio is that they work really well as a team. And maybe if you take a step backward, to identify them, we had to run for about 4 years, what is arguably the largest prospective proteomic study ever to be conducted of the human response to acute infections. And start with a host of multiple tens of thousands of proteins bioinformatically, and then down selected this eventually to 3. And these 3, while none of them is perfect in itself, they cover one another's blind spots. So let's go one level deeper. When we went on this, one of the things we were surprised to find out that as a clinical community, we've been obsessed with the bacterial side of the equation. Every biomarker that you have in 21st century medicine, 20 and 21st century medicine, has been mostly predominantly upregulated in bacterial infection. Procalcitonin, bacterial infections. CRP, bacterial infections. White blood count, bacterial infections. Absolute neutrophil count, which we use as part of routine day-to-day care, bacterial infections. What about the viral side of the equation? We couldn't find even one that was used or cleared by FDA as part of 21st century medicine. The recent FDA clearance, we actually just cleared the first viral-induced protein ever to be cleared by FDA. And so we went on this fishing expedition, and 4 years into the process, again, this was 2009, 2013, we identified this trio. TRAIL is a protein that shoots up in your bloodstream when you have a viral infection, whether it's a common influenza A, influenza B, parainfluenza, or corona. And it has this very unique property that it goes down when you have a bacterial infection. Why? Nobody really understands the reason, but it really creates a very dramatic fold change because of this up and down type of a response. And the story there's a lot of interesting stories around TRAIL, but one of the mechanisms by which it does that, it causes the cells that are infected by viruses to commit apoptosis, cell suicide, and by that protect their brethren cells. So that's one of the mechanisms that the body is using. The second one is called IP10, which is an interferon gamma-induced protein, basically shoots up in your bloodstream if you have a general infection, and more so if you have a viral infection. It recently got a lot of headlines in the context of SARS-CoV-2 and hyperreactivity of the immune response. It's also associated with lung injury, but a really interesting biomarker that plays a critical role there in clearance of infections. The third one is called C-reactive protein. It's been around for about 40 years, goes up in bacteria. And the nice thing about them, they work as a team. So as I said, they're not perfect. So take, for example, CRP. It's reasonably okay after 48 hours. But because it takes it about 48 hours to reach maximum level. But in the early phases, you have a blind spot. Whereas TRAIL at time of symptoms onset, it's already differentiated. So they cover one another. By the way, we didn't identify this. The computer identified that. This is a lot of insights that we had in hindsight when we were looking at this.
Yeah, that was going to be my next question, which is, the heart of this show is always like, artificial intelligence and its whole basket of tools and biology. So how does machine learning come into this process? Is there some corpus of training data that shows that certain levels of these 3 proteins correlate, or can you tell us, how you develop that part of the system?
Absolutely, and I think again, I was teaching machine learning at the Watson Institute over a decade ago, before it was a sexy topic. People today are using the term machine learning and data science so often, so frequent. I think what's important to say is that machine learning is part of the component technology, but there's hardcore immunology and molecular biology. So it's not just one technology that I would say it's a very high entry barrier because of that, and adds to the complexity. So that's one thing, just to put machine learning in context. Where machine learning plays an important role here is 2 places: in the development and in the final product. In the development, there's a process of how do you find the optimal team of biomarkers that would give you the best performance. And over there's a lot of activities around using publicly available data sets and proprietary data sets and data analysis and statistical analysis and feature selection and finding the right models, et cetera, coming up with what is the right model. Some of these are more conventional tests. Some of these are more cutting edge tests. In the final product, it basically uses what's called a supervised learning approach, which basically means the following. Imagine every problem, and I'm going to be a little bit technical here. Imagine you have, let's say, one feature, say a viral biomarker, trail. High levels, viruses; low levels, bacteria. You find some sort of an optimum cutoff that separates between the 2. It was the most informative biomarker that we found. Is it good? It's reasonably good, but there's no perfect biomarkers out there. We don't have them, nor do we believe they exist, nor do we believe in unicorns, even though my daughter is trying to continuously persuade me that there is one. Then you add another biomarker to that. So imagine that you have right now a 2-dimensional grid. And now suddenly every patient is mapped to this 2-dimensional coordinates, and you have a cloud of bacterial and a cloud of viruses. And you find a line that separates between it. And then a 3rd dimension, and a 4th, and so forth and so on. And eventually the problem becomes, how can I find this hyperplane or hypersurface that separates between the cloud of bacteria and the cloud of viruses? This is the essence of the machine learning. And the way you go about this, you train. You give it a lot of patients, a lot of data, and then you train the system. And the more data you have, the smarter it becomes. The same principle applies for doing diagnostics in oncology, spam detection, computer vision, and what have you. It's the same underlying, often similar underlying principles to try to solve many of these problems. So hopefully I was able to simplify and somewhat exaggerate how this is actually working and where the AI plays here.
So what's that accuracy rate of the diagnosis from your system? And is there certain things, let's say, it's good at, and certain things it's maybe not so good at?
Yeah, absolutely. So if you look at the overarching population, if you look at our pivotal FDA study, the AUC, the area under receiver operating curve on the entire population, was 0.9 to 0.97 across different cohorts, which is considered very high. So that's the short answer. The more, I would say, deeper level, there's obviously nuances across different populations. One of the things you have to show, demonstratively, what happens in children versus adults, upper respiratory tract infections, lower respiratory tract infections, et cetera, et cetera. So we've shown a relatively consistent and robust response. That's how the system was developed. But there are, for example, certain viruses that we know that we perform less accurate. For example, adenoviruses. Adenoviruses are notoriously hard to treat well. They're, by the way, they're one of the most prevalent, for example, viruses in children. Why? Because the immune response looks like a bacterial infection for many, many reasons. So white blood count is going to be elevated, procalcitonin is going to be elevated, CRP is going to be elevated, and we're often going to overtreat with antibiotics. So if you look at our performance in that particular subcohort, our performance drops somewhat from a 0.90 something to maybe 0.89. But that's actually one of the viruses that we see the most added value, because compared to standard of care, it's many times close to flipping a coin. So even though our performance is eroded in this particular virus, the standard of care in this particular situation is particularly challenged, and it's almost, again, 0.5, 0.6. So that's one example. There's multiple examples. We've been studying the immune response to pathogens for almost a decade now. This is just one interesting anecdote. And I think just connecting this to the who cares question that you had. So here's an interesting case that we had a few weeks ago. Child, young, 3 years old, coming to a major medical center, not really sure if it's a bacterial viral etiology, ran a PCR, came positive for adenoviruses. And it looked a little bit bacterial, but yeah, okay, adenovirus, that explains everything. About to release home, got a 99 score, 99 probability bacterial infection. So they started to do an additional follow-up, and then eventually turned out to be a peritonsillar, a bacterial abscess on the spinal cord of that particular child, had to be mechanically removed. And this is a very dramatic case. This is one of those potentially underused cases that could be very dramatic. This is very rare. It doesn't happen often, but again, it's hard. It's really, really tricky to distinguish bacterial viral infections. And we added this right now. This is how everything maps together to the adenoviruses and why we think this could be helpful.
So, like I said earlier in the show, you got FDA approval and granted 510(k) clearance back in September, which, congratulations, that's a huge step. But for everybody listening, what gates does that open for you? What's the pathway to getting the device out into the market?
So, as you said, first of all, you have to get the clearance, which I think took us almost 5 years of working with FDA. FDA, by the way, we've worked with them extremely collaboratively, and they've been instrumental in helping us form and shape. What's the methodology to actually prove something like this? We didn't talk about this, but how do you prove that absence or presence of bacterial viral infection in the absence of a true gold standard? So let's put that thing aside. We were able to work with FDA and come up with a methodology to do that. So now, what is required to take it to the next step? There's several things. The first one, you need, and we didn't talk about this, you need a way to measure those biomarkers. You need a platform.
Right.
One of the challenges that we had is that in the early days, none of the big strategic players, the Roches, the Abbotts, the Diasorians of the world, were willing to bet on this because the risks were so high. As you alluded to in the beginning, the graveyard, and nobody got FDA clearance, so they were not willing to bet on us. Today, some of them have become partners, and we're working with them, so it's there's been great development. But at that time, it was really hard. The platform is also a challenge because some of the proteins are picogram, some are nanogram, and some are microgram per ml, which poses, again, a challenge from a technological perspective. Again, not going too much into the technology side, but we've been able to come up with a technology or a platform that can actually measure multiple proteins across almost a 6 to 9-fold dynamic range. And so you have to have a platform, and can do that in about 15 minutes. Right now, Eran working on whole blood. The second thing you need obviously manufacturing capability, which is, again, a different story. You have to be manufacturing the cartridge. The third thing you need is building the clinical evidence beyond. FDA is great, but you have to create what's called a clinical utility, real world evidence, what have you. Working with the payers, working with partners, working with clinicians, working with the societies, publishing, building a commercial team, which we're right now established a commercial team in the US. So there's multiple things, and probably last but not least, this is too big of a challenge to go at it by yourself. You need to have partners, whether it's governments. The US Department of Defense and European Commission have funded this heavily and have been amazing partners. Whether it's strategic partners, you can't take it by yourself. B versus V is not one market. It's markets. You have patients in the wards, in the EDs, in the urgent cares, physicians' offices, retail clinics. No single player has enough of a presence in one platform that covers it all. So again, we've announced about a year ago the first partnership with Diasorin, which has today almost 1,000 install base across Europe and the US, and these large automated immunoassay machines. And that's covering certain parts of the market that are overlapping, that are complementary where we're going at. So that's a little bit of what needs to be done. But again, it's changing the boundaries of what we've been doing so far, and that's always a challenge, but also an opportunity.
So you guys raised, I believe it was 93 million, if I remember the number correctly, in new funding, which sort of really adds to the firepower of being able to take that next step. But can you envision a future where we get a solid diagnosis and an appropriate treatment plan, quickly while you're in the doctor's office?
Oh yeah.
Okay, yeah. That was the shortest answer you've had so far.
I thought you could ask something much more radical. I think there's several things that are happening. There's two major discontinuities. There's a technological discontinuity. There's a regulatory discontinuity. And I'll actually add another one, which is there's a psychological discontinuity. The technologies that we can do today, that we have today at the tip of our fingers, can provide so much valuable information that can help make better decisions. The regulatory framework has changed because of COVID. It's basically shattered a lot of things. And from a psychological perspective, I think there's a push to polarization, right? Both decentralization and centralization at the same time. And so I definitely see that happening. I think we can take this several steps further. How can we take it from physician's office, also retail clinics, and even further? And that will take time, obviously, because we're dealing here with some pretty deep questions. But I think the world across multiple fields, and this is not different than anything else, I think is definitely going in that direction.
Yeah, no, I mean, I was going to, looking at what you've created and obviously first getting everybody on board, but then seeing how it can be deployed at a CVS or something like that. It could, you could have a dramatic impact on how we manage patients, the whole antibiotic dynamic, and maybe even relieving stress on the system so that, it doesn't get overwhelmed by what your system may be able to sort of help get to a much faster, much more accurate answer to.
I wanted to say relieving stress from stressed mothers and fathers, but yeah, I agree with you. Also and by the way, as you start going from more central to decentralized, there's obviously additional workflow challenges. How do you make this simpler? There's regulatory bars that you have to meet. How do you go from a mod complex to a CLIA waived a test that can actually go to those directions? So we have work. There's work to be done, but I think we've been able to potentially break a glass ceiling in terms of getting the clearance. And now I think with that, there's going to be additional innovation that will come both by us and others, right? They're entering the space.
So just slightly moving to one side is like, how has MeMed responded to, say, COVID-19? I think you guys have developed a test that runs on your platform and predicts how severe the infection will be. How does that test work? Like, did your previous work, and also did your previous work like on the platform prep you for this virus? Or I'm just curious how it works, and how you got there.
Absolutely. So it always starts with a clinical question. I mean, many of us are technophiles, but at the end of the day, it's about solving a real problem. And the problem here is defined as following. You have, say, a SARS-CoV-2 positive patient presenting, say, to the ED. And one of the questions that we have in mind is whether that patient is going to deteriorate or not. Do we escalate treatment or not? And it's a real question, right? And the more, the more stress the system is feeling because of the peak of a pandemic, the more important that is. So we said, well, how can we harness the technologies, the framework that we created, host response in general, the biomarkers we've developed, the platform that we have, the biobank and what have you. And how can we take a process that maybe took 10 years and now collapse it to something maybe that's 10 months? How do you get a 10x? And first of all, with amazing partners around the globe, we start running huge clinical studies to basically collect patient samples. We also use, again, information from the public domains, our own repository, our own previous data, because from many perspectives, SARS-CoV-2 is very interesting, but guess what? Similar to SARS and to other types of severe viruses, there's differences, but also commonalities. So we use a lot of the bioinformatics, the previous data samples, current data samples, the know-how, and the platform that's readily available right now that can measure basically anything to collapse this and develop this product that just got clearance in Europe that basically allows to take a snapshot of the immune response again in real time and give you a risk stratification regarding the probability of a patient to experience a severe outcome defined as ICU level of care and mortality within 2 weeks. Again, it's only clear right now in Europe, not cleared in the US. And we view this also as a stepping stone going beyond just COVID severity to a general severity signature. So what if you could do both B versus V and severity? And what if you could do it in the same, same cartridge or what have you? How could that? So I think what's really interesting, we built here this core technology. We went after one big problem, B versus V. But now that you have that, you're like a child in a candy store. There's many more things that you can do. And rather than taking you a decade, you can start collapsing things because there's a lot of resources that you can now leverage or platform that you can now leverage. So that's the story around me with COVID severity.
Yeah, platforms are wonderful in that way, right? That I like a platform more than I like a, sharpshooter bullet from an investment perspective, thinking about it that way. But so you recently got COVID. We were supposed to talk like over a week ago, and I we had to postpone it. Did you use the test on yourself? I mean, if you did, did it work the way you thought it was going to?
Yeah, so yeah, I got COVID from my daughter. We went on a trip, and 5 out of 5 Eden family members got infected. So yes, it was, at least from our small experiment, it was very infectious. We got the Omicron. Actually, we didn't have symptoms, apart from the fact that I think it just jacked up the energy level of my kids. So before we talked, I told them running around the house, and thank God, my wife didn't kill any one of them. So there's no casualties from this infection. So because we didn't have any symptoms, we didn't go to the ED. It was not relevant. You have to have SARS-CoV-2 symptoms. So in that case, no need. I mean, we were pretty much honky-dory. But what I can tell you is that on the B versus V, again, it's potentially bacterial versus viral infection is potentially the most prevalent indication in children. And my children, those little incubators of bacteria and viruses, are no exception. So I had a chance to use the technology on my kids many times, including last time was about a month and a half ago on my eldest daughter. It was before going to a business trip, and my wife is asking, is it a B versus, is it a bacterial viral infection? I said, I don't know. She's telling me, the shoemaker is going barefoot, so we ran it. It was a viral infection, no antibiotics, went back to school. So, and I got a lot of brownie points with my wife and my mother-in-law, which is obviously always very, very strategic. That's probably
no, that's a good one. Yeah,
you have to be in good terms with your mother-in-law. Exactly. So we're actually using this quite often in our families as well, and it's very gratifying.
Excellent. So now you guys are, I believe you have an office in Haifa, which I remember as being beautiful and hilly and wonderful food, and then you have Boston. What are the strengths of being in these two locations? What happens in Boston that can't happen in Haifa and vice versa?
So first of all, again, we're going after a global problem, and you have to have a global team with global perspective. Whatever you have in San Francisco today, you have tomorrow in Shanghai, and the day after that in Tel Aviv. So you have to look at this from a global perspective, number one. Number two, since the US is the primary market, I said we have to build a very significant presence in the US. Why specifically Boston? Very talented a pool of, a pool talent that's very wide in the domain. There's a big overlap in terms of hours between Boston and Tel Aviv, so you can grow one unified team. And that's where we're basically building our US headquarters. And the team is quite complementary. Again, we've recruited by now roughly 25 to 30 folks with a very strong background, both IBD, Troy Battelle, formerly Thermo Fisher, was running commercial for microbiology in the Americas. Fred was running corp dev for bioMérieux, again, another large multinational. Jim Catherine was former head of sales for BioFire. Again, I'm not sure from the audience was familiar with the name, but and so forth and so on. And Will Harris was running our marketing, global marketing, again, ex-Amazon, and then before that, 15 years in IBD. So it's really bringing here a blend of, we call this affectionately an anti-disciplinary team. We don't care about disciplines. We care about solving big, ugly problems. And so you have to bring the IVD experts with the clinicians, with the folks on the big data science side or and the molecular immunology and the manufacturing. And no single location has all the know-how, and no single location has the recipe, because frankly, we're doing here something new. There is no real tech like this. And so bringing this pool of talent, I think has really helped us propel us moving forward. And it is the bridge to be able to launch in the US. US very focused commercially, marketing product, where a lot of the, I would say, more of the molecular immunology data science team is more in Israel. I'm simplifying and exaggerating, but that's that's some of it.
So the last funding round, was that the argument to the investors, like we need to hire these types of people to help blow this out? What was the rationale for that last round?
So basically three things. Number one, commercialization. US, Europe, Israel, that's our initial focus, and then the rest of the world. Second is product pipeline. So we talked about bacterial versus viral infection and a bit about COVID severity. But what would you do if you had a blank canvas and these platforms to go after the immune response and measure the immune response? What additional big problems would you go after? So it turns out that there's some pretty interesting stuff, and we're working on additional activities. So that's the second thing, product pipeline. And the third thing is scaling manufacturing. So that, as I think people have a new appreciation for manufacturing and supply chain during COVID times, it's a really big topic and critical for success. So these are the three major elements that we raise the funds for.
Yeah, no, sounds. I've seen this rodeo a few times, so yes, I understand completely. So, well, especially because I come from the diagnostic world, I can't wish you enough success, because we need more products like this out on the market to help us manage patients and help give physicians better information so that they can make better decisions, right? More informed decisions than just, looking at a patient and trying to figure out what's going on. So I wish you incredible success, and I'm great to have you on the show.
Thank you so much for the kind invitation. Enjoyed our discussion.
Thanks.